Good afternoon, Professor Tom Zoeller will present his lecture entitled The Brain on Endocrine Disrupting Chemicals. Pioneer in the study of the thyroid hormone in brain development, Professor Zoeller will discuss the importance of thyroid function in pregnant women and concerns about the industrial chemicals to which we are all exposed. Professor Zoeller has been an active member of the campus community since 1994. He served on multiple personnel committees at various levels, has advised research students at all levels, and has served as chair of the Department of Biology. Professor Zoeller also spent a year as a visiting professor of anatomy at the University of Otago School of Medicine in New Zealand. The internal and external recognitions that Professor Zoeller has received while a member of our faculty clearly demonstrate the esteem in which his colleagues, both at UMass and his field generally, view him. He was awarded the Samuel F. Conti Faculty Research Fellowship in 2007, was recognized nationally as Scientist of the Year by the Learning Disabilities Association, and received an Individual National Research Service Award from the National Institute of Neurological Disorders and Stroke, among others. As an expert in the role of thyroid hormone in brain development, Professor Zoeller has reviewed for numerous journals and publications and funding agencies, and has been invited to speak in many national and international venues. We're delighted to have Professor Zoeller here to speak with us today. I'd now like to invite his chair, Professor Ralph Kostrom, to the podium to introduce our distinguished faculty lecturer. Ralph.
Thanks, Jim, and thanks everyone for coming. It's a great turnout. It really is a pleasure and an honor to introduce Tom for this distinguished faculty lecture. A little farther back in his history, Tom just told me he was in the Army and was drafted in the Army and came out of the Army and ended up getting his degree at Indiana University, his bachelor's, and he said he could have gone anywhere and he wished he'd known at the time that somehow he ended up in Indiana, but I don't know what that says about IU, but I think it's a good place. He ended up with a master's and PhD degrees from Oregon State University in Corvallis and did postdoctoral work at NIH at the National Institute of Neurological Disease and Stroke. Tom joined, as Jim said, joined the UMass faculty in 1994, having come from an assistant professor position at the University of Missouri and a medical school there. In fact, Tom has been a mentor of mine since I joined the faculty and has provided advice and support for which I am largely grateful. I think the one possible exception is his advocacy for me as chair. But over the last 3 years, I have started to forgive him. I continue to appreciate his advice in the administrative realm as well. So my job here is to give you a little introduction on Tom's work and how it actually led to this honor. I guess I'll break it down into two aspects that I think help get your head around it. The first is the obvious basic research aspect of his research, and the second is how it's merged into the science policy world. So in terms of basic research, Tom's contributions has been fundamentally important in the area of, and very productive. He's built a research program here that has 107 publications at his CV, which is, I think, was a year old at that point, so there's more than that now. I looked up his H-index impact factor over the years, and it's over 30, which is one of the highest I've seen when I've looked up these impact factors. And something the administration, and now including chairs, tend to like to hear about. He's brought in almost $6.5 million in research funding since he's come to UMass. So the basic area of research, of course, is endocrinology, the field trying to understand how remarkable things, these little hormones that float through our bodies, affect everything from physiology to behavior. I think anyone who has a teenager or can remember being one can attest to the power of hormones. Not only do they affect our own functions that we see like that, they also affect, it turns out, embryonic development. And Tom has provided or contributed fundamental information about how hormones, in particular thyroid hormone, affects development of the embryo. So he was the first, actually, to show 12 years ago now that thyroid hormone from the mother actually affects the development of the embryo, of the fetus. Now that was a big deal because of the dogma at the time was that thyroid hormone could not get across the placental barrier, and so could not affect embryonic development. So over the last decade and a half, this has now become the new paradigm in the field, and it's actually been important in that it's opened an entire new area of research and changed the standard of care for pregnant women. Because of his findings, he was awarded Scientist of the Year award for that fundamental finding from the Learning Disabilities Association of America. So in terms of applied research then, this work really merged into public policy in the 1990s when Tom was selected to participate in the Endocrine Disruptor Screening and Testing Work Group of the Endocrine Society. So the committee, he says, met every 5 weeks for a year and a half. And that experience led him to realize that basically the way we assay endocrine disruption, how we evaluate the risks and regulate the risks, were simply not adequate. And so he's been part of a series of papers that focus originally on polychlorinated biphenyls, PCBs, and other environmental chemicals, and the broader theoretical issues of how you actually determine whether something is dangerous and whether we need to regulate it. So his work has shown that endocrine disruptors can interfere directly with thyroid hormone function, bind to and mimic the thyroid hormone receptor, and have big impact on the development and function of mammals. So I think that's really an important illustration of how basic research can be translated into fundamental new understandings that then guide public policy. Okay, so the most recent manifestation of this is actually pretty stunning. He's now a member of the Scientific Advisory Board for the Environmental Protection Agency. He has played a huge role now in testifying in front of Congress on endocrine disruptors. He's joined efforts by the UN and the UN Environmental Program and World Health Organization to review the state of science of endocrine disrupting chemicals. And a new document was launched by the UN, WHO, in Nairobi last Monday, I guess. So this is current, and it's high impact, and it's international in scope. So just to sum up, over the last 2 decades, Tom has established himself as an international leader in a fundamental area of research that is critical importance to human health: the role of thyroid hormone in development and the effects of endocrine disrupting chemicals on human development. Obviously leading to his talk title, The Brain on Endocrine Disrupting Chemicals. So with that, thanks.
Well, thank you very much for that generous introduction. Thanks to those who nominated me and also for the selection committee for giving me this opportunity to speak about something that is going to come up here in a second. So what I want to do is really talk about the work that I've done kind of in broad strokes. I'm not going to go through a lot of data, but just kind of tell you what the issues are and kind of conclusions we've made. And then I really want to switch the talk to why I think the regulatory system is not adequate and what we might be able to do about it. So to reiterate some of the introduction, when I was at the NIH, I really went there to study neuroendocrinology. That is, how does the brain control endocrine systems? That's really what I was focused on. I worked on reproductive systems, but also the thyroid gland. So I was beginning to think about how the brain controls thyroid function. There were really 2 events that caused me to move into the field that I'm working on now. Now, the first is becoming a parent. I think everybody who becomes a parent, there are many mixed emotions, but one for me, maybe this sounds kind of geeky, but I really started to think about how hormones control development. Development is really this issue of how does a single cell become a human being with personality and sense of humor and perception. Because I was thinking about thyroid hormone, and I knew thyroid hormone is essential for brain development, it really got me to think about how does thyroid hormone control early brain development. And you know, this was in the 1980s, early 1990s, and online searching was just beginning. And I can remember doing the first kind of lit search through Medline, and there was not a single paper on thyroid hormone and brain development in the fetus. Now, I'm a scientist in today's world, and the first thing that meant to me is it's got to be fundable. So, so I figured that this is a good place to begin. The second kind of experience that led me to this is this experience on the Endocrine Disruptor Screening and Testing Advisory Committee. Now, this committee was designed to advise the EPA about how to go about evaluating chemicals for their ability to interfere with hormone action. I'm a thyroid guy, and so that meant thyroid hormone action. If we don't know what thyroid hormone is doing, we can't evaluate whether a chemical interferes with it. That was my guiding principle through the year and a half of these debates, but we didn't have the data. And as a result, what got set in kind of an ironclad way was a mechanism to evaluate thyroid disruption, in quotes, that just simply won't work. And even today, today the EPA is kind of wrestling with this issue on a number of levels, but the, the kind of things or rules set in stone are already set, and it's probably going to take a long time to change. And so I've been, complaining about that. So what I want to do is really kind of talk about at the beginning, why do we think thyroid hormone is important in development? What's the evidence for that? And what has my lab, which really, this is kind of the Amway plan. When I say my lab, it means all the people that worked in my lab that I get to take credit for. So, and one person I'd like to point out right away is Ruby Bansal, who has been in my lab for 12 years and really has been an instrumental part of everything that I'm going to talk about today at one level or another, either directly publishing papers or indirectly in terms of providing kind of intellectual support as well as logistical support. So why do we think thyroid hormone is important for development? There's really two kind of historical stories that tell us this. The first, on the left, is frog metamorphosis. So frogs won't become, tadpoles won't become a frog unless thyroid hormone is present. And this has actually, even today still, tells us a lot about how thyroid hormone works, what's the mechanism, and it's relevant to humans. But the second story is Cretinism, and we've known about Cretinism for about 400 years and known that it's related to goiter, which is in areas of endemic iodine insufficiency or deficiency. The thyroid gland doesn't work, the mother doesn't make thyroid hormone, and as a result, there's very severe mental retardation and retardation of stature and growth. Now, this is a situation of severe thyroid hormone deficiency and severe effects. But there's also a condition that's kind of more random that occurs in the population. It's not a genetic situation mostly, but when a baby is born that doesn't make thyroid hormone for any number, there's probably 5 or 6 reasons for that. Thyroid hormone levels are low, and it can have a very significant impact if it's not evaluated and treated early, within really a couple weeks. And so there's a system in place where all babies born are evaluated for thyroid function. And you can imagine how during the 70s and 80s, when the screening program kind of became available, that physicians really wanted to know how to optimize treatment for these babies. What that literature tells us is that very small differences in thyroid hormone levels in the baby has a permanent effect on cognitive function as that baby grows. In fact, even one study that was a prospective study for over 20 years demonstrated that verbal ability, verbal IQ, at age 22 was related to thyroid hormone levels at age 2. So it's not just right immediately after birth, but thyroid hormone is important for development during this whole period. Now, all of these studies are really focused on the postnatal situation. And it really was dogma on the basis of very few studies and little evidence that thyroid hormone didn't cross the placenta. And in those days, I think in those days, like the late 80s, early 90s, which really doesn't seem that long ago for some of us. But in those days, it was kind of considered that the placenta was this sort of this wonderful thing that made sure that all good things went to the baby and nothing bad went to the baby. In fact, it was even thought that alcohol didn't get to the baby, which seems kind of odd chemically. But so when I first began to think about this during the EdStack committee, I really wanted to focus on how thyroid hormone can affect the fetus. And this is important because the fetus in a human, the fetus doesn't begin to make thyroid hormone until the second trimester and really doesn't make very much until about week 20. By that time, brain development isn't complete, but it's certainly well on the way. And we knew from a number of studies that the brain contains all the machinery for thyroid hormone to work, but we had no evidence that it works or what it's doing. So how do we approach a system that we don't know anything about? How do we begin to experimentally move in there and kind of inform ourselves about what's happening? So really the way we approached this is to use what we knew about how hormones work. The first issue is that, like all hormones, thyroid hormone exerts its effect by interacting with a specific protein called a receptor. If you don't have a receptor, then the molecule that we call a hormone can't have this hormonal activity. So the receptor itself is what is the business end of the endocrine system. We also know that, in fact, I was in a lab at the NIH where the guy who initially cloned the thyroid hormone receptor also worked. And so I was kind of privy to a lot of unpublished information about thyroid hormone receptor and what its structure was, et cetera, et cetera. So we knew that thyroid hormone receptors are there. We also knew that they're nuclear receptors. And these receptors bind to DNA and regulate gene expression. Okay, so that was our hook. What are those genes that thyroid hormone regulates? If we knew what those genes are, they would tell us, kind of just by the nature of the proteins that are being made, what kind of developmental events are being affected by thyroid hormone early on. In addition, each one of those genes would represent a marker of thyroid hormone action that we could use to determine whether or not chemicals interfere with thyroid hormone action. So we could kill two birds with one stone or get 2 grants to study the same kind of thing. Okay, so this was actually the first student I had, the first grad student I had in the lab, Amy Dowling, came in, and halfway through her project I started to get nervous because I had no idea that this was going to work. And when you put a student on this and you see how hard they work and their career is dependent upon your sort of scientific acuity, also called luck, it makes you nervous. So it turns out, though, that she published a number of studies and won the Byron Prize for, which is a kind of best dissertation award in the MCB graduate program for the work that she had done here. And she was absolutely spectacular in this work. We used what we now think of as kind of a primitive technology for genomics. So it was differential display. Anybody that knows what that is knows kind of how primitive that is now. We've got much more sophisticated technologies, 15 years later. But the basic principle of it was that in a way that wasn't that was very open and non-biased, we could identify genes that are regulated by thyroid hormone in the fetal cortex before the fetus can make thyroid hormone. We could manipulate thyroid status in the mother and identify these genes in the fetal cortex. And so we published a series of papers on this, which gave us a lot of hope that we were actually on the right track. And the conclusion from that really was that thyroid hormone from the mother can affect gene expression in the fetus. Some of these genes that were most kind of interesting to us were genes that cause cell differentiation. What that means is early in development, a cell hadn't decided what it wants to be when it grows up. And thyroid hormone comes in and sort of coaxes it to move in a certain direction, to become a nerve cell or a supporting cell or an oligodendrocyte that makes myelin, etc. So thyroid hormone in that way sort of is a morphogen in the sense that it causes cells to be different from what they are. Now that's a problem from a strategic point of view, because when, the thing that differs between two different kinds of cells, a liver cell, a brain cell, are the genes that it expresses. So if we're using genomics as a way of identifying what thyroid hormone does, then what it means is that many of the genes that are different in a cortex that has low thyroid hormone versus high thyroid hormone are probably just related to different cellular phenotype. The cells are different in the brain between those two situations, and if we just characterize those differences, it's not telling us exactly what thyroid hormone is doing. So the question is, what are the direct effects of thyroid hormone on brain development and on gene expression? And it was not until actually maybe 5 years ago or so that there were technologies available that would allow you to do that. And so I'm going to go through this just kind of in a broad stroke way because it's really cool technology. And I like that stuff. So remember the thyroid hormone receptor, what's called TR here in blue, is bound to DNA. And we want to know what that DNA is. So what you can do is take the cell and covalently, chemically link the thyroid hormone receptor to that DNA. And once it's stabilized on that DNA, you kind of blast the whole thing apart. You shatter the cell. You fracture all the DNA into small pieces. Then you take an antibody to the thyroid hormone receptor and fish it out of this big mix. And when you fish it out, it brings that little piece of DNA with it. And then what you do is spread that DNA that we've now isolated that's only bound to TR, you spread it over this plate that has the entire mouse genome printed on it. And it'll stick to the pieces of DNA on this plate that is similar. And of course, you pay a lot of money for this stuff. And they've got software that essentially tells you what the gene is, what the DNA is, and everyone in this green thing down on the bottom, each one of these spikes that comes up above the horizontal line is a place that is specifically, that is enriched. That's kind of the language, but it means that you fished out that piece of DNA and you can point to that gene as a direct target of thyroid hormone action. So what we came up with are a series of genes on the right that are direct targets. At least the data that we've got suggests this. We could be wrong. I'm wrong a whole lot, so it doesn't bother me to be wrong. But some of this is right. It'll be up to somebody else to figure that out. So when you look at what these genes do, first one is cell fate specification. That's cell differentiation. So thyroid hormone in the early cortex is telling cells what to become when they grow up. If the mother doesn't have enough thyroid hormone, those cells in the fetal cortex are not going to be what they should have been. Now that seems to me like a problem that we need to protect against. If there's a chemical in the environment that interferes with this regulation, that's going to be a problem. Okay. So the question is, can industrial chemicals interfere with this hormone action? Now, this is a picture of thyroid hormone here, and it's not a very good one. And I don't have a marker, so I'm going to walk away because you need to think about certain things here. Thyroid hormone has these two rings. There's one ring here, and here's another ring. The purple is iodine. So it's an atom on there that's big and it's negatively charged. This red thing is an oxygen atom that's in between these two rings. So these rings can bend like this around the oxygen. They can also spin. Okay, so these iodine atoms causes those rings to be in a non-coplanar conformation, okay, because they're negatively charged. It's like if you take two magnets and put the two negative poles together, they repel each other. That's what's happening here, and that's what forces this molecule to look the way it does in 3 dimensions. Okay, so now we've got polychlorinated biphenyls. You can see these same 2 rings. Now, these things can spin, but they can't bend. So when I first looked at PCBs, I thought, you know, they're not going to bind to the receptor because they've got to have that, the molecule has to have this flexibility to fit in there. I was wrong. So chlorines are also negatively charged, and they also force these rings to be either in a coplanar or a non-coplanar conformation, very thyroid hormone-like. Here's polybrominated diphenyl ethers are flame retardants. And these guys have an ether link between those two rings. So not only does it spin, but it also flexes, which is even more thyroid hormone-like. Here's triclosan, which is in your toothpaste and, you know, biocide soaps and all this kind of stuff. If you're going to make something that looks like thyroid hormone, that's it, because it's also hydroxylated. It's got this ether link, it's got these 2 phenyl rings, it's got chlorine atoms that hold it in a position that looks like thyroid hormone. Now this is bisphenol A. You can't really see it in here. A friend of mine it was a long time ago, I think it was probably right at about the end of the 90s or early 2000s, he sent me this picture, and he's a computer guy. And he was doing these docking experiments where he took the thyroid hormone receptor binding pocket and was fitting it with different molecules, and he found bisphenol A fit perfectly. And he sent me this thing and he said, you know, you got to take a look at this because bisphenol A is becoming big news. It looks to me on the computer that it binds to thyroid hormone receptor. And I said, well, you know, bisphenol A is an estrogen. I don't want to, you know, work with estrogens. So, so I didn't do anything, and a couple years later, the Japanese published a whole bunch of papers on how this thing binds to the thyroid hormone receptor. And, you know, it's another one of those, you know, really great choices I made. In each one of these cases, you can see the structural similarity. Everybody in this room is contaminated with each one of these chemicals. Every baby born in this country is contaminated with each one of these chemicals. So even though I'm not showing a whole lot of chemicals here, they're important because we are all contaminated in an unregulated way. So what did we do? The first experiment that I attempted was to, whoops, I'm telling you now why we looked at PCBs. Why did we choose polychlorinated biphenyls? The answer really is that we knew a lot about it. People have been studying it for a long time. Their production has been banned, but they're all over the place. They were used in a whole bunch of different, I've only got a couple things here. They're industrial settings for anybody who, like me, who went to grad school or undergrad in the 70s. You know, oil immersion lenses used Aerochlor 1254, which is a pure polychlorinated biphenyl mixture. The optics were great on these things. They were indestructible. They were, you know, heat insensitive. You could do all kinds of stuff with them. They also didn't go away. They were persistent in the environment, not completely indestructible, but very sturdy. I think the half-life in the environment is in the decades to 100 years, something like that. And of course, they bioaccumulate through the food chain, and that's why we have a lot of them. So their production was banned by Congress. It wasn't banned by a regulatory action. There was no risk assessment that said PCBs are bad. And this is a critical issue for me because I think even after the 40 years of studying PCBs, human studies, molecular studies, animal studies, etc., we know these things are bad. Nobody would defend bringing PCBs back. And yet, they would make it through the regulatory system just the way they did in the 1960s and 70s. So we're still contaminated. They're still relevant. The FDA actually has maximum tolerance limits for PCBs in animal feed, in dairy milk, and you know, this a little, it's not disingenuous, but it's a little bit playing with the numbers. I could make the case that the doses of PCBs that we give animals and show effects in the brain are actually higher than the maximum tolerance limit for dairy milk. So it's still a relevant issue. This is just a review of PCB levels in a variety of matrices like maternal fat, placenta, fetal fat, cord blood, breast milk, etc. There are a lot of studies of PCBs in breast milk, and these levels can be very high. This top level at 1770, this is an average number for that particular cohort. There are geographic differences, but largely we're all contaminated. There's also a number of studies in humans, largely kids, and it turns out that PCB exposure before birth affects brain cognitive function, and that cognitive function is manifested in many ways, including lower IQ. In this study, they were looking at these percent of commission errors. So this is a measure of impulsivity in young kids, and they linked not only PCB exposure to measures of impulsivity and low IQ, but also structure of the brain. And this part of the brain labeled A, we know is affected by thyroid hormone. So it begins to pull together this idea that PCBs can affect brain development in humans, and this was actually done in DC. For anybody that's from DC, you might think about that. But it also brings in, I know somebody here from DC, so it also brings in that there's a structural, potentially a structural change that's related to this interference. This is a review that just shows a lot of studies looking at cognitive function in kids as a function of PCB exposure. And you can see in the third column there, all of these things, all of these arrows pointing down. There's an enormous amount of concordance in these kind of epidemiological studies linking PCB exposure to cognitive function in kids. This is kind of a review of studies that have looked at PCB exposure and thyroid hormone levels, and you can see over in the right-hand side here, the arrows are going all which ways, and there's not so much concordance. So it doesn't look like there's a lot of agreement in studies that PCBs cause a reduction in thyroid hormone levels, which then may affect brain development. Now, our studies may explain why that is the case. This is a paper from, or this is a figure from our first paper on this. And what we found, paradoxically, was that PCBs could increase the expression of genes, of a gene, single gene. We looked at a bunch in this paper, but I'm just showing you one here because the picture actually is pretty cool. It's kind of colorful. And it's showing you the cortex of a developing rat brain. And PCBs activates the expression of a gene that we know is regulated by thyroid hormone. So it suggested to us that among this mixture of PCBs, there are some that can bind to the thyroid hormone receptor and activate gene expression. Now, it's not that simple, but I'm going to pretend that it kind of is. And what I'm going to focus on, kind of instead of going into all the biology, is to focus on a pathway that's really kind of interesting that we've figured out first in cell culture. And that is that some PCBs can bind to and activate a gene in different cells, largely the liver, that this gene CYP1A1 is an enzyme. And that enzyme is supposed to help purify things that we eat. So we eat something, it goes to the liver, and this enzyme is one of a bunch of enzymes that's supposed to take care of bad things. Okay, now instead of taking care of bad things, what it does is modify different PCB molecules. And that modification actually is necessary for this PCB molecule to bind to the receptor and activate gene expression. So your liver is doing what it's supposed to be doing, but instead of deactivating the chemical, it's actually activating the chemical. And this two-step process, we have proof in culture. So in cells that we put in a dish, we can figure this out because we can completely control CYP1A1 activity. We can do all this kind of stuff. We've got very clear proof that this two-step phenomenon is occurring in cells. Well, we can't prove it in an animal, but all of the data are completely consistent with this kind of two-step model. And we've also got evidence in a fairly large 170, it's actually about 200 mother-infant pair cohort from which we obtained placentas. So we took the term placenta and we looked at kind of molecular markers of this kind of two-step mechanism, and the data are completely consistent. So from cell lines to animals to humans, we see this same kind of two-step phenomenon. Now this is important because in part this enzyme CYP1A1 is induced by smoking. It's also induced by a lot of other things. So one way that people might become more or less sensitive is by the other kinds of things in their life. If you smoke, you're going to increase your likelihood that PCBs could be acting as these kind of direct endocrine disruptors. In addition, it explains why interfering with thyroid hormone action isn't reflected by changes in thyroid hormone levels in the blood. So when you just do a blood test for thyroid hormone, it's not going to reveal what these chemicals are doing. And so they're kind of a little stealth system, and trying to identify that in a large screening system is going to be very difficult. So I'm going to move on to this argument that despite certainty that PCBs are neurodevelopmental toxicants, and nobody doubts that now, and strong evidence that at least part of this neurotoxicity is related to thyroid hormone interference, the endocrine disrupting screening program today would not pick it up. And so why do I think this is true? So I'm going to switch now from our data to just giving you some of the logic. So this is the kind of the first, wave of defense for the endocrine disrupting screening program. This is tier 1, so all pesticides but no other chemicals have to go through this system. There's two stages to this. The first is in vitro, which just means in a test tube. And there's estrogen receptor binding, receptor transcriptional. I don't see anything that's related to thyroid hormone there. So that part's not going to catch anything that's related to the thyroid hormone. The in vivo component are animal studies. All of these animal studies have been around since the 30s or 40s. When people were thinking about how to, really even what are we worried about in the human population, in the 30s and 40s, they were worried about different things than they're worried about today. 18% of children today in the US has a diagnosis of a neurobehavioral disorder. 18%. The Uterotrophic Assay is a rat assay that looks at uterine weight, among other things, but mainly organ weight. The Hirschberger assay was developed in the 1930s. That's looking at male reproductive organ weight, mainly organ weight. The pubertal rat assay for male and female, this is the only place where there are thyroid hormone endpoints in a mammal, in this whole screen, in this whole system. essentially that is set up to look at whether or not chemicals can interfere with puberty. You're kind of seeing there's a trend here. It's all reproduction. Not that that's not important, but I grew up Catholic. So the only endpoint of thyroid hormone here is thyroid organ weight and thyroid histopathology only if organ weight is changed. Now, how do we think about organ weight, and how is that predicting neurotoxicity or any other kind of toxicity? And so I've got this analogy here. Let's say you've got a really cool car, and you bring it to me because the engine doesn't start, okay? And so what I do is pull the engine out and weigh it. Now, how many people think that you're going to feel really good about me as a mechanic? Now, it gets worse, though, because what if the engine starts fine, but the back two wheels don't turn? What do? I pull the engine out and weigh it. And really, this is exactly what this EDSP does. It's one size fits all. They've got one set of assays that they apply everything to. Okay. Now, just to prove that this is relevant, I went to the AAAS meeting in Boston about a week ago. There was a session on Saturday afternoon on bisphenol A. So this guy, Barry Delclose, is from the FDA Food Safety Division. What they're proposing in this, in this series of talks is that bisphenol A cannot be bad for people because the dose that we're exposed to, the amount of bisphenol A that we're exposed to, is far below what causes an adverse effect. Okay, so this is a rat study they're talking about here. And I've kind of highlighted this area for the results. And you can see here bisphenol A affected gestational weight gain, so that's body weight, and had multiple reproductive organ, and what they mean by that is organ weight, but also some histopathology that they did. And they also looked at some clinical chemistry, which probably isn't part of the kind of guideline studies, so they were really pulling out all the stops here. And their conclusion was that the internal dose of bisphenol A causing these changes in organ weight and body weight are about 1,000,000-fold higher than what humans are exposed to. And then at the end they say continuing efforts to detect more subtle effects of bisphenol A at lower doses are underway. Now, I'm actually part of that sentence. Now, the thing that I pay attention to is that organ weight is considered adverse, but changes in the structure of the brain is considered subtle. So, in other words, the first thing is the measurements that they're taking don't map to the epidemiology studies that link bisphenol A exposure to cardiovascular disease, to diabetes, to behavioral changes in girls. And those behavioral changes are largely masculinized play behavior. So this is more aggressive play behavior, et cetera, et cetera. So they also, I knew because I'm part of that study, I know that brains from the animals that he presented data on had been evaluated by a faculty member at NC State and had shown that there are changes in gene expression in regions of the brain that are known to be sexually dimorphic at the lowest dose of bisphenol A. So this was done in a completely blinded way, and yet he didn't mention that. It doesn't fit with the story that bisphenol A exposure is so much higher than what the effect is. And I don't think it's disingenuous. I think it's a genuine belief that organ weight reflects toxicity, not changes in structure or function of the brain. And I have to go back to kind of my early days of working on car engines, and I don't think that engine weight really is reflective of function. It doesn't seem that difficult. So just to give you kind of move out of this kind of myopic view and think of it in a more global sense. First of all, the US produces or imports an estimated 73 billion pounds of chemicals a day. That's a lot of chemicals. We are being exposed to many of these. There's about 80,000 chemicals licensed. In the US, the WHO on this writing group that I was working with estimated about 140,000 chemicals licensed worldwide. Many of these are produced in small quantities and are really not an issue. In this country, the estimate is about 3,000 a year are produced at 1 million pounds or greater, which is high production volume. Chemicals wind up being in places that you don't expect. For example, the paper wrapper of hamburgers are sprayed with the chemical that makes Teflon. So these are perfluorinated chemicals. These are interesting chemicals because nobody has ever been able to measure any degradation in the environment. So as far as we know, they're with us forever. They won't degrade. And yet that's what's sprayed on the inside of a hamburger wrapper so that you don't get grease on you. Also, plastic diapers are another kind of classic case where there are a number of chemicals put in there largely for preservatives. A lot of times these things are completely unregulated. Nobody knows who regulates plastic diapers. It's not FDA food safety. It's also not necessarily EPA. This final picture is of flame retardants. These are polybrominated diphenyl ethers. We have data on this, but I'm not going to really show that. We see many of the same features that we see with PCBs. I've also got a study in collaboration with a colleague at UCSF. California, for a long time, has had this fire retardant policy or rule that requires the use of a lot of these brominated flame retardants. And babies born in California have levels of polybrominated diphenyls that are higher than anywhere else on Earth. It's amazing the levels of these chemicals that are found in babies in California. That's not why they're blonde and tan. We don't know anything about the fact or how these chemicals might interact. What I've just shown you and what we study in the lab is the way in which chemicals act like drugs. They might be weak drugs, but they're drugs that don't go away, and they're drugs that are given to everybody regardless of their developmental stage. We don't know how these things are going to interact as a mixture. The director of the National Institute of Environmental Health Sciences, Linda Birnbaum, is kind of famous for saying that we live in this chemical soup, and one of the problems with this is we can't definitively go into a human population and say that chemical does this thing, because you've got 50 or 60 or 80 or 100 chemicals that you have to contend with, and you're not going to be able to figure this out. So it's a very difficult problem to prove that a chemical is producing, that this chemical is producing the bad thing that you're looking at. Also, this study really affected me also, because what they did was to take 10 random samples of cord blood from a geographic diverse area in the US that were given by the American Red Cross. They measured all the chemicals that they could find, and they found 287 chemicals in just 10 cord blood samples. About 45 of those chemicals were the same in all 10 babies. So when I presented this at a congressional briefing once, the American Chemistry Council got up and said, just because these things are there doesn't mean they're bad. We've got really sophisticated techniques, and so therefore we're finding lower and lower levels of these chemicals. But my argument is that what it means is that we as a society think it's okay to put 45 or 100 chemicals in every baby born in this country until we find out unequivocally that it's causing some bad effect. And that bad effect had better be reproductive organ weight. Okay. So the Endocrine Society also has developed a couple of policy statements about this. In 2009, they published their first scientific statement on endocrine disruption that concludes that this is a very significant public health issue that needs to be addressed, and we followed this up last September with a paper that really articulates why we think the way we have to approach identifying endocrine disruptors needs to be changed. We need to use principles of endocrinology and not principles that are not related to the way these chemicals act. In addition, with a writing group of about 16 people that was sponsored by the UN Environmental Program and WHO, we published a paper that was just launched a week ago that Rolf talked about, that essentially says that this is a global threat. There's greater and greater information, first of all, about diseases in the human population globally. So there's a shift. There has been a shift over the last 50 years from communicable diseases to non-communicable diseases. So the incidence and prevalence of non-communicable chronic disease is higher in the world than it has ever been. And we continue to ignore things that we can do to try to address that issue. The UN finally passed a resolution at the International Chemical Management, I forget, it's ICCM3, I forget exactly what it stands for, but it was in Nairobi, International Conference on Chemicals Management. What this essentially does is require the UN to educate member nations about the issues of endocrine disruption, and in fact they have partnered with the Endocrine Society to provide experts to the 5 different regions of the UN to begin to kind of educate policymakers from countries within those regions. So I'm going to end there and just kind of thank the people in my lab and show you that we don't just stay in the lab, or they don't just stay in the lab and do things. This is actually in the Swiss Alps at a Gordon conference, and to the left is Jintao Wang, who was, she's from Chengdu University and was a visiting professor here for a year. Stephanie Gira was a grad student who's now at Harvard. And Ruby Bansal, to the right, is a person in my lab that has been working for about 12 years. These are additional grad students, all of which have gone on to nice places, like Sohee Yu is now at Penn. Dave Charlin is a faculty, just took a faculty position at University of Minnesota after a postdoc at the NIH. And Kelly Gallagher, who also won in fact, both Dave and Kelly won Byron Prizes for the MCB, and she's a scientist at PVLSI. So I think I'll leave it there, and thank you for your attention.
We have one more rather important element to this afternoon's proceeding, and that is that on behalf of Chancellor Subaswamy, I want to present you with the Chancellor's Medal.
Thank you very much.